Hello everyone, I'm Dibyendu Samanta from IIT Kharagpur, and I'm discussing about the course Introduction to Complex Biological System. Today, this is the fourth topic, Introduction to DNA Replication. So, during the last class, we mostly discussed about DNA structure, mostly chemical and physical properties of DNA. And today, I am going to discuss about how the information of DNA get copied so that the information can pass from one generation to the next generation. So, this is the DNA replication. So, in this topic, I will concentrate on basic understanding about DNA replication, followed by one classic experiments commonly known as Mesenchel and Stahl's experiment that proves the basic understanding, the basic mechanism of DNA replication. So, now if we see, like particularly I discuss about the differences between DNA and RNA, its chemical composition, everything. So, DNA like deoxyribonucleic acid. So, here like most of those nomenclature, they are very much pre-logical, they have some kind of logic. So, as I explained that deoxy means that 2' carbon of that sugar, like oxygen atom is not there, that is why deoxy. And then ribonucleic acid, the sugar is of pentose type, particularly ribose, that is why deoxyribo, then nucleic acid. Acid because whatever we discuss based on that, if you see the backbone of the DNA that comprises of the sugar phosphate repeat. So, because of this phosphate group, DNA is negatively charged as well as it has acidic property. So, nucleic acid. But then what about the nucleic? So, this is because of if we discuss little bit about the discovery of DNA, then we will understand the name nucleic acid. So, now many of us like we think that DNA was discovered by Watson and Crick. So, that is not right, because almost 80 years before the discovery of DNA structure, which was done by Watson and Crick. So, DNA was discovered by Friedrich Misser from white blood cell. So, what he observed, that he isolated some substances from the nucleus of those white blood cells, and those are very acidic in nature. So, that is why this is called nucleic acid, some acidic substances found in the nucleus, nucleic acid. And now I already explained that deoxyribonucleic acid. Similarly, ribonucleic acid, so that 2' position, still it has the oxygen group, the OH group is present there. So, now just I will start today's discussion, basic understanding about DNA replication. The major important thing from Watson and Crick's discovery is their specific base pairing, because if we go back, as just I told about the discovery of DNA, similarly after that many scientists contributed a lot to understand the overall features of DNA, like how they are covalently linked, the phosphodiester bond, and how that nitrogenous bases are attached to the sugar, all those things were known. And then what is the major thing, important thing of Watson-Crick discovery is this, the specific base pairing, this double helix structure, that A always base pair with T and G always base pair with C. So, this is the most interesting part of Watson and Crick's discovery, because that actually tells us that how the information can go to the next level. That means how the information can be copied to the next generation. So, that is, I would say, the most important thing. So, here in that paper particularly, so this one Watson and Crick published this paper in Nature in 1953, and in this paper at the end they mentioned that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is very important, because before that how this information is going to the next level was not known. But here if you see, like this is one DNA structure, as you can see that it's run from 5 prime to 3 prime direction, one strand, and the other strand 5 prime to 3 prime direction, because DNA strands are anti-parallel. And as you can see that A always base pair with T, AT, GC, it's going like this. Now there is some advantage of this. Also I discussed during the last class that, for example, if you increase the temperature of a solution where you have DNA, that those two strands will be separated, because this hydrogen bonding will be disrupted. As a result of that, those two strands will be separated. So if we can separate those two strands of DNA, then again we can copy the information, because if you know just the sequence of one strand of this DNA, then you can automatically put the other like sequence of the other strand, because this is always a very strict rule that A base pair with T and G base pair with C. Those two strands are complementary in nature. So as a result of that, this DNA, if I write in this way, so 5 prime A, T, C, G, T, C, A, 3 prime, exactly whatever mentioned here on the left hand side. So as a result of that, the complementary strand should be 3 prime, T, A, G, C, A, G, T, and 5 prime. But here this is double stranded DNA. They have hydrogen bonding between this A, T, G, C, all those things. So now if there is some mechanism so that these two strands are getting separated, then we can synthesize the new strand because of this complementary rule. So as a result of that, in the next stage, if these two strands are getting separated, 5 prime, A, T, C, G, T, C, A, 3 prime, plus this 3 prime, T, A, G, C, A, G, T. So these two strands, then we can actually make the new strand again from here. So here T, A, G, C, A, G, T, 5 prime, 3 prime. This underlined strand, this is the new strand. And here also the other strand will be synthesized from 5 prime to 3 prime direction. So as a result of that, you are getting A, T, C, G, T, then C, A, 3 prime. This underlined strand is the new strand. So as a result of that, if you see, this is the template strand, and then we are getting the new strand. So as a result of that, one strand is the old strand here. This is the old strand or the parent strand, and this is the new strand. Similarly here, this is the old strand or parental strand, and this is new strand. So now we have 2 DNA molecules, 1 and 2, these 2 DNA molecules, and in each case you have one strand is new and one strand is the old strand. So here in this DNA molecule, for example, that 50% of DNA is conserved, 50% old and 50% new. So that's why this like model is called semi-conservative model of DNA replication. So this is semi-conservative model of DNA replication. Semi-conservative model of DNA replication. So as you can see in this figure that those two strands So here those referred in blue color. So those are the parental strands. So if we have some kind of mechanism so that those two strands are getting separated, then you can see this new strand. So in pink color here. So those are the new strands. So as you can see, this is semi-conservative replication. So here in this DNA, you have one old strand and one new strand. Similarly, in this DNA also, you have one old strand and one new strand. But during this time, this is proposed by Watson and Crick, that semi-conservative DNA replication based on their DNA structure. But the thing is, at the same time, there are some other hypotheses of DNA replication. So like I would say this is one semi-conservative replication. Then the second one is conservative mode of DNA replication. So conservative DNA replication. So based on this hypothesis, so they are suggesting that if you have this one just a double-stranded DNA, now by somehow after replication in the next round, what you are getting that this DNA is preserved. This is the old DNA plus one new DNA is being synthesized here. So this is both strands here new, and here both strands were old or the parental strand. So as a result of that, the parental DNA is conserved here, and the both strands containing the new strands in this DNA. So that's why this is called conservative mode of DNA replication. And there was another mode of DNA replication hypothesis, that is dispersive. That means that is suggesting that somehow this double-stranded DNA, they get small break in both strands, and from there the new strand will be synthesized. So as a result of that, in the final molecule, that means from one DNA to the next cycle, when we will get two DNA molecule, both the strands will have the new and old strand mixed together. This is called dispersive mode of DNA replication. So dispersive mode of DNA replication. So now we need to come out with some experiment so that it can be proved that which one is right. Although Watson and Crick suggested based on their solid data that this could be semi-conservative, but after a few years, like Messenson and Stahl, they did some experiment, Matthew Messenson and Franklin Stahl. So this is commonly known as one of the most beautiful experiment in biology because of the simplicity of the experiment, but still clearly demonstrate that DNA replication is semi-conservative. So this is commonly known as Messenson and Stahl's experiment. So first I need to explain the background of the experiment, that few things I need to discuss so that we can explain quickly about the details of the experiment. So what they did, so they work with E. coli. So E. coli, I would say E. coli is bacteria like prokaryotic cell. So in E. coli cell, we have circular DNA, just one circular DNA. So this is E. coli and DNA present in E. coli. Now, E. coli cell with proper environment, with proper media, it can get doubled. That means from one E. coli, you will get two E. coli cells almost in 20 minutes. In 20 minutes, E. coli will divide into two. Now, that means by that time, the DNA of this E. coli or the chromosome of this DNA chromosome here, same thing. So that is getting replicated, and both like these two E. coli cells, they are getting DNA. So they work with E. coli cells. So now, in order to understand what is the mechanism of DNA replication, So we have to somehow divide, like we have to somehow understand, or we have to somehow designate which one is the new strand and which one is the old strand. We have to differentiate at the end after the experiment. So that was the major thing in this experiment. What Mesanson and Stahl did, so they grow E. coli cells. So what happened? So they took E. coli cells and they grow in media. So we grow E. coli cell generally in liquid media. So this is called LB, liquid media. So they made some media where they put nitrogen 15. So nitrogen source is just N15. So not the N14, it's a heavier isotope, N15. So they made some kind of media so that you don't have N14 in that media, only you have N15. And similarly, they are also growing E. coli cell in another flask where they have only N14 containing media. So they added ammonium chloride where the nitrogen is either N14 or N15. So as a result of that, as we already discussed, that nitrogen is an important atom in DNA. So during replication, this nitrogen will be incorporated in DNA during replication step. Now, after going several hours, they extracted DNA from this bacteria which were grown in N15 media. Similarly, they extracted DNA from those bacteria which were grown in N14 containing media. Then they analyze on some ultracentrifugation. So just simply if I want to say that if this is centrifugal axis here and here you have two tubes here, two tubes. So this is the centrifugal force. That's why when it is spinning very rapidly, those tubes will be like this. But when we stop the centrifugation, then those tubes will be just like this, just like this. Because of the centrifugal force, it is spreading in this way. Now, what they did, they loaded DNA sample like from this N15 sample say here and from N14 containing sample in this tube. So in this tube they have some solution which is made up of very high density cesium chloride. So they particularly use cesium chloride during this experiment, but many other things can be used. So cesium chloride, very highly dense cesium chloride they have in this tube. And now they loaded this DNA which was extracted either from N15 or N14. And then they started this centrifugation for a long time. So this is called equilibrium density centrifugation. So as a result of that, if you spin it for a long time, then since you have very dense solution in the tube, then because of the centrifugal force, DNA will migrate in this direction, right, in this direction. But also you have very highly dense solution. So as a result of that, the DNA will move based on their weight. So now N15, since it's heavier, so I would say if N15 is present here, they notice that the N15 containing DNA moved towards this direction more compared to N14. So those are same size tube. So as you can see here, this is the centrifugal force. So they notice that this is N14 containing DNA and this is N15 containing DNA. So that means when DNA is getting replicated in presence of N15, all those nucleotides, they have the nitrogenous bases and all those nitrogen are heavier nitrogen, N15 atom. So as a result of that, overall the molecular weight of those DNA, those are heavier than the N14 containing DNA, and that's why they migrated more compared to the N14 containing DNA. So this is the basis of their experiment, so that they can basically distinguish between the two type of DNA strand, the N14 containing DNA strand and N15 containing DNA strand. Let's come to their experiment and discuss about that. So here what they did, so they took E. coli cell, and they grow this E. coli in N15 containing media. It's a liquid media, N15 containing media for long hours, for example for overnight, and then they harvested those cells. So cells were grown for very long time, like I would say 10 to 16 hours, and then they harvested E. coli cells. So they harvested E. coli cells. So how they will harvest? So that means again they took the whole media where you have those E. coli cells, they spin it through some kind of centrifugation method. So as a result of that, E. coli cells are heavier, so that they will settle down at the bottom, and now we have to remove the supernatant, so that we are actually trying to remove any solution that contain N15 media. So now you have just cells without the media. Now few cells, or I would say a fraction of those cells, were taken to, like, they isolated DNA. DNA was isolated from these cells which were grown in N15 media overnight. But the rest of those cells they transferred into N14 media. Rest of the cells they transferred into N14 media. So now they wait for 20 minutes. So they transferred here in this media. So I would say here that they transferred here, and you have N14 media in this flask. And then they took sample. So here, after transferring into N14 media, they took sample at 20 minutes. 20 minutes later, they took some cells and then they extracted DNA. So they extracted DNA here after 20 minutes. And then still you have cells in the media. Then again they took cells at 40 minutes. Again they isolated DNA. And then another sample 60 minutes, and they isolated DNA. So now we have 4 DNA samples. One was here, so here, which was directly harvested from cells which were grown in N15 containing media, followed by we transferred those cells only into N14 containing media, and we waited for 20 minutes, and then we have taken another DNA sample, we have extracted, and then after 40 minutes again we extracted DNA, and after 60 minutes again we have extracted DNA. Now when they analyze these 4 DNA samples through the same centrifugation technique which I discussed in the last slide, so here I am just drawing 4 similar tubes here. Four similar tubes. They are not looking exactly similar, but you just imagine that they are exactly same tube, and all of them, they contain cesium chloride. And they loaded this DNA sample in this tube, and what they did, they just centrifuged for a long time, whatever I discussed during the last slide. And they observed that in this first tube, here, this tube number 1, in this tube, they found that they have the DNA band somewhere here, they settled here. This is the DNA. So I already explained that since these E. coli cells were grown overnight in media containing N15, so these are very heavy DNA, right? All of their nitrogenous bases are labeled with the N15 atom. So very heavy DNA here. Now, in the second tube here, after 20 minutes, what they found that DNA is present little bit at a higher position, not exactly at the same position where you have in first tube. And then, interestingly, in the third tube, which were taken, those DNA was taken after 40 minutes. So now this DNA is present somewhere here and here. So see, in tube number 1, like 100% DNA, they are heavy. That's why they migrated maximum. So this is the centrifugal force in this direction, right? So as a result of that, they migrated maximum. Now, in tube number 2, if you see, they again get a single band, but this is little lighter than this tube 1. So as a result of that, something, a different species here, not completely heavy, not completely light. This is some kind of hybrid DNA, and 100% here, 100% hybrid DNA. Why hybrid? Because see, in the next tube, you have here this hybrid DNA, which is 50%, and this is the light DNA, again 50%. Why light DNA? Because this is, migrating minimum. So this is the light DNA, and based on their density, they could conclude that this is 50% light DNA and 50% hybrid DNA. And now, in the next tube, they found that here only you have 25% and here 75%. So 25% hybrid and 75% light. So this is light DNA, that means DNA containing N14, and this is hybrid DNA. So after getting this result, actually it completely ruled out, or I would say it's completely convinced that DNA replication is semi-conservative. Because now if I draw here, this DNA, both the strands are labeled with heavy atom, heavy nitrogen. That's why they are heavy. But now, if DNA replication is semi-conservative, then what will happen? In the next round of replication, we have one strand which heavy nitrogen, and this is the new strand with dotted line. Similarly, here new strand with dotted line. So as a result of that, this DNA now hybrid. They have one heavier strand, one light strand, and they are settled here. So as you can see here, 100% hybrid DNA. This is because of semi-conservative. If it is conservative, then what we would say? That again we will see some are completely light and some are completely heavier DNA molecule. And in the next round of replication, here we would see because now we don't have any more N15 containing DNA. So here we have 4 DNA molecule. Again here you can see this one with one heavy strand and one light strand. This one heavy strand, one light strand, and here both these 2 DNA they have both strands are light. So as a result of that, 50% DNA here, they are hybrid, and 50% DNA, these two, 50% DNA, they are light. So these experiments together, it proved that DNA replication is semi-conservative. Then why we are taking sample after 20 minutes? Because I explained that in favorable condition, E. coli, they get double in 20 minutes. So as a result of that, we could say that this is the, just after one round of replication, you have sample here in tube 2, and then another round of replication in tube 3, and the next round of replication tube 4. So the same thing, just we are showing from some images from some textbook. So as you can see, this is the original or the parent molecule. Both strands are labeled with N15, and here the first generation, both strands are hybrid. So that's why you can get this hybrid band here. And similarly here the second generation, as I just explained, so you have two strands, two DNA molecule which are hybrid, and two DNA molecule which are completely light. So all those, this is kind of classic experiments, and many scientists believe this is the most beautiful experiments in biology, and it's very simple experiment, but it clearly demonstrates that DNA replication is semi-conservative. That's all for this class. And for further reading, you can refer any standard textbook, including this Lehninger Principle of Biochemistry by Nelson and Cox, or Essential Cell Biology by Alberts.